Hi everybody. In this unit we'll be introducing our first functional system, which is the central nervous system. It consists of the brain and the spinal cord. As you'll see though, I'm going to spend most of my time talking about the brain. I always like to preface this unit by mentioning that I'm not a neuroscientist, and brain science is complex, it's a rapidly evolving field, and there's a pretty steep learning curve. So my goal for this unit isn't for you to become an expert, it's really just to lay a foundation that you can build on in your future courses. In fact, if anything I say in this unit is particularly interesting, the P&B department has courses dedicated to the biology of the brain and to neuroanatomy specifically, so you can take those if you're interested. I'm going to start by talking about how the brain is both physically and chemically protected. I'm going to talk a little bit about cerebrospinal fluid, or CSF, about the blood-brain barrier, and then I'm going to jump into talking about some specific structure-function relationships that we see in different parts of the brain. I'm going to wrap up our discussion of the brain by talking quickly about the process of learning memory, and then we'll talk briefly about the spinal cord, because we're going to follow up talking about the spinal cord in class when we're together. So, our brain is contained within the cranial cavity, and it's physically protected by a number of things. Some of them are listed here on this slide. So superficially, you have your skin, which creates a physical barrier with the external environment. If we go a little bit deeper to the skin, then we have the bones that form the actual cranium. You know that many of those you've already learned about in the laboratory portion of the course. And the nice thing is that you can double dip, because the naming of the cranial bones often intersects, a little bit at least, with some of the functional brain regions that we'll be talking about a little bit later in the unit. Deep to the cranium, we have meninges, which protect both the brain and the spinal cord. And then lastly, we have the interstitial fluid of the brain, which is known as the cerebrospinal fluid. That provides both chemical and physical protection. So, of those things that I've just mentioned, the two that are probably the least familiar to you up to this point are the meninges and CSF. And so we're going to talk a little bit more about those two. I want to quickly elaborate on the meninges, which again are responsible for physical protection of the tissues of the central nervous system. There are actually three meningeal layers. The outermost layer is known as the dura, and it's made of fibrous connective tissue. So this is actually the strongest meningeal layer. And it actually consists of two layers. There's two layers to the dura. And between those two layers, we can find a lot of the blood vessels that supply the tissues of the brain. So again, Grey's Anatomy fans, you've probably heard the phrase subdural hemorrhage before, which is when someone has a rupture in the blood vessels within that dural space. And usually that happens in response to head trauma. It can be quite severe. The middle meningeal layer is known as the arachnoid layer, and it's made up of epithelial tissue. And it's an important landmark for us because cerebrospinal fluid is located in the space beneath the arachnoid layer, in a region known as the subarachnoid space. The innermost meningeal layer is known as the pia. This is a really thin layer of cells surrounded by a thin layer of connective tissue, and it sits directly on top of the brain. It follows all of the underlying architecture of the brain, sort of like a layer of plastic wrap would. It actually goes all the way down into the gyri. Now, the cerebrospinal fluid is the interstitial fluid of the brain. So given what we know about from our membrane potential unit and from the neurophysiology unit, the composition of CSF can directly impact the function of neurons in both the brain and the spinal cord. Most of the cerebrospinal fluid is produced within structures in the brain known as ventricles. You can think of them essentially as a CSF storage reservoir. You can see the ventricles pretty clearly in this image in the upper right-hand corner, and you can hopefully appreciate that the ventricles are quite large. Now, having fluid inside the brain creates some buoyant forces that help to reduce the weight of the brain, as opposed to, for example, having a really densely packed tissue. But CSF isn't just stored within the ventricles, it's also circulated by the ventricles. So CSF can move from the lateral ventricles, which are on either side of the brain, into the third ventricle, which is pretty centrally located. And from there, it can travel through a structure known as the cerebral aqueduct into the fourth ventricle. And from the fourth ventricle, it can go through the central canal into the spinal cord. Now, this is actually a really important point because it tells us that CSF is continuous between the brain and the spinal cord. And that means that it has a relatively uniform composition. And that's why when physicians are interested in knowing the composition of the CSF, like for example, to look for infection, they'll often do a spinal tap, which samples the CSF from the base of the spine instead. So this spares the patient the need to have a needle stuck into their brain. This is a lot less invasive. But since our focus right now is on the brain, I also want to point out that CSF can travel into the median and lateral apertures, which will eventually deliver it into the subarachnoid space. And from there, it can be circulated throughout the rest of the brain. Now, if you look down the lower right-hand corner of the slide, you can see there's a zoomed-in view of this region. The CSF that's circulating through the subarachnoid space is shown in black. And that's going to allow it to travel all through the brain, and it's going to facilitate exchange with the tissues of the brain. But eventually, the CSF drains out of the subarachnoid space into the venous blood. You can see that being represented by the arrows in that diagram, and the venous blood being represented by the blue space. So we're moving from black into the blue space in a single direction. And the reason that it's in a single direction is because it's happening by bulk flow. The subarachnoid space is at a higher pressure than the veins, and so the CSF follows that pressure gradient. Now, about 75% of the CSF is produced inside the ventricles by a specialized type of epithelial cell known as an ependymal cell. The ependymal cells form a tissue that's known as the choroid plexus, and again, that's found inside the ventricles. So from this picture here on the left-hand side, you can see this compartmentalization pretty clearly. The ependymal cells create a physical barrier between the blood vessels that supply the brain and the CSF. And again, ependymal cells are a type of epithelium. They're actually transporting epithelium. So as you might expect, they're pretty selective about what they allow to pass from the blood into the CSF. Because of this compartmentalization and selectivity, the CSF and the blood have different solute compositions. This is pretty interesting because up to this point, and in every unit after this, we kind of always make the assumption that the plasma and the interstitial fluid are very, very similar in terms of their solute composition. But the central nervous system is an exception. CSF, the interstitial fluid, has a lower concentration of protein, of glucose, and potassium, and it has a higher concentration of sodium. Having that higher concentration of sodium allows us to create the big concentration gradient that we need to make it favorable to bring sodium into the cell during the action potential. And having low extracellular potassium in the CSF allows us to have continuous potassium efflux that we know is important for maintaining negative resting membrane potential. So there's a reason that the solute composition has to be different between those two compartments.